Hello, and welcome to Week 15, Module 15A of Biology 240 Micro. Today we will be talking about acellular pathogens. And this image and slideshow is kind of brought to you by kind of OpenStax. I will be using materials from the lecture. The first slide here is basically showing you a little bit about viruses. Now, we've been kind of fighting outbreaks forever, and COVID is just kind of one of those viral outbreaks that kind of went further than any of them did before. Ebola is a hemorrhagic disease that's caused by a virus. In 2014, we saw the first large-scale outbreak of Ebola. These occur every couple of years. In fact, one occurred this year, but we had a lot of other things to kind of worry about besides Ebola. Again, with the advent with the electron micrograph, we're able to kind of see pictures of these guys. Classically, viruses were first studied looking at plants, specifically looking at the tobacco mosaic virus. This virus was able to be crystallized, and it did not grow on itself. It needed plant material to grow. This was funded by the tobacco industry. They had a lot of money. They have something that's called a golden leaf. I'm in North Carolina. When I was in North Carolina, I saw a little bit of that. They are very generous towards research. Another virus that we look at, and we know a lot about materials, are by basically looking at these things that are called bacteriophage. And this is an electron micrograph of a bacteriophage. This is a virus that infects bacteria. Phage basically means to eat. And this illustrates, this is a drawing of this bacteriophage right here, one of the first ones. It kind of looked like a moon lander, but basically it has for attachment, these structures for attachments. And so in the last class that we were looking at diseases as one of the stages of a pathogen is to attach to its host cell, and how these viruses attach are these kind of proteins that are right down here. We want to look at sizes. Again, 1 micrometer is about the size of a mitochondria or of an Escherichia coli. Typically, viruses are much smaller than this. Hopefully, they're going to be a little bit larger than some molecules and proteins, but they're going to be smaller than a bacteria. And so we're talking something that's maybe around 50 nanometers for kind of the small virus. The larger viruses like smallpox are going to be almost in the 10th of a micrometer. And again, when we're looking at like red blood cells, 5 micrometers, plant cells, 50 micrometers, etc., they're size that are relatively small. So we need to know what those are. Again, we have different types of viruses that were kind of dependent on their structures. Adenoviruses use spikes made of glycoproteins from a capsid that bind to the host cell, and so that these form the attachments that are on here. The enveloped immunodeficiency viruses uses spikes made of glycoproteins to embed its envelopes to its host cells that allow. And so again, these are the spikes that are in here. And COVID has a spike that we use. Again, these viral capsids, depending on how they're produced, can be helical or polyhedral, or they can have a complex shape to do that. So we're looking at this through the electron micrograph. And so again, the tobacco mosaic virus is an example of those kind of helical structures that you basically produce a protein that can attach on kind of two sides, and it will actually form this helix, and you'll have your nucleic acid material inside of there. The polyhedral would be an example, would be the human rhinovirus. And then a more complex shapes basically would be made of fragments of the envelope as it's being reduced. Now, we want to look at kind of the life cycle of a phage, and what we call a virulent phage only does what we call the lytic cycle. And so what's going to happen is this phage is going to attach to your bacterial cell. It's going to penetrate and get into the bacterial cell. It's going to synthesize all of its material. These materials would then come together in a process that we call maturation, and then we have lysis. And so basically what happens is you have this pulse, and that you may see no activity for a while, and then all of a sudden you see 1,000 bacteria. So for one, sorry, not bacteria, but viruses. So for one virus, you would would see a pulse of these 1,000 viruses. What we call a temperate bacterial phage has both a lytic and a what we call lysogenic cycle. And so in this one here, I'm going to go over the lysogenic cycle. So it's not always in the best interest of a phage to actually kill its host by producing more phage. You know? And those phages can kind of hang around. And so what we happen is in the lysogenic cycle, what we see here is the bacterial phage gets into there, and instead of producing more phage, the actually phage incorporates into the host genome, and then as the host divides, so does the phage. And then under stressful conditions, or conditions which are going to see the death of the cell, what can happen is the phage can produce a whole bunch. And we kind of see something a lot like this in, let's say, cold sores. So where people either, when they get sunburnt or when they're under stress, they start to have these colds developing on their mouths. And so this figure 6.9 just basically shows you the mechanism of what we call specialized transduction. And specialized transduction, what's happening is that every once in a while these phage actually get an aberrant piece of DNA, and they can incorporate that DNA. When that DNA gets injected into the cell, it can get incorporated. And what happens is sometimes these phage, that there's no viral DNA in there, it's only this specialized transduction genetic material. So that's an interesting way. And again, we're using that. So the adenovirus, the adeno-associated virus is one of the viruses that we actually use to actually, we produce this virus and inject it into people. It's one of the vaccines that we have, not the Moderna or the Pfizer vaccine. Those are mRNA vaccines. So this is slightly different. But again, it's something that we do use. Again, influenza virus is one of the few RNA viruses that replicates in the nucleus of the cells. In an influenza virus, the glycoproteins attached to the virus to the epithelial cells, and as a result, the virus is engulfed. And so what we have here, and so here's the influenza. It goes inside. We see penetration. We see uncoating of the material. In biosynthesis, we basically have the materials that are produced inside here. We have assembly outside of the nucleus. These, again, the nuclear material, the genetic material is made inside there. The proteins are made outside, and then we see release of this material by budding, that kind of burst that we talked about. When we talk about RNA viruses, we can actually have a plus strand, single strand RNA that can be directly read by ribosomes. This essentially looks like a messenger RNA to a cell. Or we can have a negative strand, which first uses that the negative strand is actually used as a template to synthesize the positive strand before it can be kind of synthesized. HIV is an envelope icosahedral retrovirus that attaches to a cell surface receptor of the immune cell and fuses with the cell membrane. The viral contents are released into the cell where viral enzymes convert the single-stranded RNA genome into DNA and incorporate it into the host genome. And so these particular HIV can kind of hang out in the genome for another for a while, and that's what we call that lytic cycle. And then what happens is the RNA is made. That RNA can then be processed into more of the virus. Varicella zoster is the virus that causes chickenpox. It has an enveloped icosahedral capsule visible in a transmission electron micrograph. It's a double-stranded DNA and becomes incorporated into host cell DNA. What can happen is this particular virus can lay dormant for a while, and then every once in a while it kind of pops up. So everyone who's had chickenpox as a child will actually have it kind of manifest kind of later on in its life. There are vaccines for this. And so what we see is, unlike the growth curve for bacteria where we have that kind of easy sigmoidal curve, what you have is basically a one-step multiplication step. And so your virus inoculates. So this is looking for the number of infectious viruses. So what happens, you put a bunch of viruses inside of a bunch of bacteria, and those viruses, for the most part, kind of disappear for a while. These viruses go inside the cell, and then what happens is that all at once we get a number of virons released. And so what you can do is you can divide the number, you get the total number of viruses that you get in the end by the number of viruses that you put inside, and you can get this average burst size. And so roughly it's about 1,000 viruses for each infection that occurs. Now, viruses are nasty things, as we all know, and so these are individuals that are working in Africa on the Ebola virus. It's basically they have breathing systems, they have protected coating, they have negative air pressure cabinet, kind of where they're working at. So it's really hard. Imagine you're working on this. You're working with something that's very scary, and then you're having trouble seeing. You're uncomfortable, and kind of everything else like that. How viruses were first actually isolated or not isolated, or identified, is that, again, when researchers were working on some plants, they had a filter, and what they did is ideally they filtered out the bacteria, but even on these small pore sizes, they had some materials coming through. And so if you remember from the beginning of the talk that these viruses are very small, and so it's these viruses that we are kind of looking at here. How do we count viruses? And these are actually tissue culture plates. We looked at these plates, or we looked at these tubes. These are T25 tubes. We looked at those when we were looking at viable cells. On here, this is a phage titer, and so we have a bacterial lawn, and these are the bacterial phage that are kind of growing in this bacterial lawn. This is a T4 phage. That looks a lot like the bacterial phage that you saw before. And so these are one of the ways that we have of counting it. How do we grow viruses sometimes? And so remember, viruses need to be grown in kind of living tissue. One of the great things that we have for growing up viruses that we kind of don't necessarily know, and so again, we may not have agar plates to grow viruses, like we said there. And so what we can do is we can use embryonated chicken eggs. And so what we have is the embryo, and viruses will grow in certain areas. So it's one of the reasons why when you get vaccinated for things like the flu, they ask you if you're allergic to eggs, because some of these viruses are actually made in eggs, and so we got to be a little bit careful with that. We can also, again, cell culture. We can use primary cell culture, and so you have cells that are growing, and so in a primary tissue culture, you take some cells from the lung and you kind of let those grow, and you can pull them, and you can only kind of pass them a certain number of times before they die off. And actually transform cells, these are cells that basically you have continuous celling. These cells will continue to grow forever. And so the HeLa cells, you may have heard about those cells, are a continuous cell line, and we've learned a lot from those HeLa cells. This is an example of how we can use those HeLa cells. And so here are the HeLa cells. Here's the extracellular matrix that they release, and then here are some viruses around those HeLa cells. And this is an image contrast using electron micrographs. And so, again, what we can, various viruses that are out there. This is a list of some of the viruses that are out there, some of the ones that emit, typically that you would see. Here's kind of what the viruses look like, and then and so we'll give those names out to you before. One of the ways of looking at viruses is that viruses, very interestingly, can cause what's called hemagglutination. And so a really easy test for that is to actually take red blood cells, put them in a sample, and then you have no reaction. In hemagglutination, you actually would have this occurring, and this is off. This is hemagglutination would actually be this little dot here, and non-hemagglutination would be where these cells are out there. And so we'll talk about that. And so again, that's NAGNAM and some of the other things that we look at for there. Another test that we can use is very similar to the test strips, the antibody test strips that we did, or a sandwich ELISA that we did. And so what you can do is you can take and you can stick a virus on a surface, and then you can have antibodies with a conjugated enzyme on them. And if it binds to it, when you add the substrate, it will change color. If it's not, then you will not kind of see that. Viruses, there are some small little viruses, and how they come about is this. In this case here, we have a virus that's transmitted by basically the knives. And so what you do is when you plant potatoes, you would cut them in little pieces to get the eye of the potato. And if you do not wash that knife in between each and every potato that you use, sometimes if you contaminate a potato, you can contaminate the rest of your crop with those knives. Now, viruses aren't the only infectious subcellular infectious particle. We have something that are called prions, which was discovered here in the Bay Area at kind of UCSF. And then basically what's really kind of interesting with prions is that you have an exogenous protein. And this protein, when it interacts with an altered protein, will actually become altered itself. Again, this protein can spontaneously alter to become kind of an infectious. And so this is looking at here at brain tissue, and these are the plaques that are formed in the brain tissue by these prions. And so here's a normal brain, and here's a brain with that. And so here's normal tissue, and here's that tissue with the plaques. And so this is something that we call mad cow disease. It's also called Creutzfeldt-Jacobs disease. And again, for a while there, we were really afraid that this disease would kind of take us over. If we go back, this is a bacteriophage, and you should know that's that. You do need to know what's going on with the lytic cycle. This is the lytic cycle right here, and so we're showing that the phage binds to it, it then incorporates into it, it takes over the host cell materials. You have your new bacteriophage form, and then you all of a sudden have a burst. And so be able to understand how to calculate that kind of burst value. This right here is showing you what we call the bacteriophage, and so you'd have the opaque area here would be the bacterial lawn, and here you have the phage growing up in here. And so it's kind of the opposite of the colonies, or it's very similar to the colonies, right? And so when you have a bacterial colony, you have the bacteria that's growing on the media and growing up in here. It's actually the cells that are being, and you can tell by the cells being damaged in here. And so I'm going to end almost all of my lectures with what do tapeworms eat? They eat you, or they eat the material that you eat. But this has been the talk on acellular pathogens. And if you have any questions, put them up on the discussion board. We'll have a week 15 discussion board that you can look at, and we will do some other activities. All right, I will catch you later. Thank you very much.